So in the last session, we have discussed about the structure of ovule or megasporangium also. So here megasporangium or ovule, so it is, we have seen, so it is the extension. So there will be a cushion structure, that is placenta. The attachment of that one to the cushion, so that is ovule will be there like this. And this, the stalk of this ovule, we call this as funicle, and the junction between ovule and funicle, this junction we call this as hilum, and this is funicle. And inside of this one, there you can see the outer integument. So this one is outer integument, and this one is inner integument. I am not drawing completely here, so that we have discussed yesterday. And inside of this one, you can see this a layer of cells, that is nucellus. So this is nucellus. Nucellus is nothing but it is a food reserving tissue, where it will be abundant protoplasm will be there. It is food reserving cells will be there. This tissue we call this as nucellus. And within that what you are seeing, that we are calling this as, that is the main female gametophyte, that is the embryo sac. That embryo sac will be present there inside of it. And this end, where it is having a pore or opening, this end is there, no? This end we call this as micropyle. This end we are calling it as micropyle, and this swollen end is there, no? This we are calling it as chalazal end. Chalaza is nothing but swollen, and this end is nothing but micropylar end. Micropylar end or micropyle it is, and it is chalazal end. And this only micropyle only after the fruit formation or seed formation. So in the seed you will be seeing micropore, no, micropyle there. So that is the same thing. And here the completely like formation of megaspore, everything we will be seeing in this location or in this place. So next we are going to discuss megaspore formation. So that is nothing but in megasporangium you will be seeing. So this is megasporangium. So in this hilum is there, funicle is there, micropyle is there here in this micropylar end or micropylar pole, which is chalazal pole. This is embryo sac, and the tissue which is surrounding to the embryo sac is nucellus. So remember, immediate next to the embryo sac there will be nucellus tissue. Nucellus tissue is nothing but it is having food reserve material which helps in development of embryo sac there. Development of embryo sac, it is done by, or the development of megaspore, it is done by nucellus here. Nucellus tissue will be there surrounding to that. This is nucellus tissue. And next to that what you are seeing is inner integument, and next to that outer integument, micropylar and micropyle is there. So outer integument, after the fertilization, outer integument is going to form testa, and the inner integument it is going to form tegmen. This, it is data of 11th grade morphology of flowering plants. In morphology of flowering plants, seed coat is there, no? Outer seed coat it is made up of testa. So inner seed coat it is made up of tegma. So those are of this one, outer integument and inner integument of ovule or megasporangium. And this type of ovule, we, in the last session I said, this type of ovule we call this as anatropous ovule. And even I said that why we are calling it as anatropous ovule. So there are many types are there. If you didn't attend the last session, you can go and watch that in a 2x speed. So next is megasporogenesis. So it is similar to the microsporogenesis here. Megasporogenesis means what? So in the same name itself it is there. Megaspore genesis is nothing but formation. Genesis is nothing but formation here. So megasporogenesis in the sense, the process of formation of megaspore from megaspore mother cell. So from megaspore mother cell through meiotic division again here, through meiotic division. So this is nothing but megasporogenesis is here. So where it will be taking place? First identify the location where the megasporogenesis will be taking. So in the last session we have seen there, so there is formation of ovule. So there is a formation of ovule was seen. So ovule is here, and this part it is nucellus is there, and within that embryo sac is seen. Okay, and this is micropylar end. Now I am taking only nucellus part here, so I am drawing the nucellus here. So it is a nucellus tissue it is, or the nucellus cells. So these nucellus cells, they are food reserve material. So it is having food reserve materials in this, and the ploidy of this one, ploidy means what? Ploidy means a set of chromosomes within a cell there. So here the ploidy of this cell, it will be 2n. It will be 2n here. The nucellus cell, the ploidy of nucellus, it is nucellus tissue, and the ploidy of nucellus is 2n. Diploid it will be. If you check this ploidy of this cell, that is also diploid. That is also diploid, because still only in gametes we will be seeing the haploid content there. So still there is no formation of gamete is seen. So just it is a surrounding of that one. It is like if you check in the microsporogenesis or in microsporangium, so there within this what you will be seeing, sporogenous tissue is there, and these are going to form microspores or pollen grains. So surrounding to this pollen grains you will be seeing tapetum there. So tapetum cells are found which are rich in food reserve material, which helps in nourishing or development of these pollen grains there. So this tapetum cells also is seen in diploid condition. So the same case here nucellus, here it is nucellus is of diploid in condition. So the nucellus cells, it is in diploid condition. So one of the nucellus from this, see there are many cells are surrounding here. So one of the nucellus from these surrounding cells will take initiative, it will come forward from this micropylar end. So usually in most of them it will be coming from the micropylar end. So there one of the cell, it will take initiative, and it will working as megaspore mother cell there. So it will be coming front there, which will be acting as megaspore mother. So it is acting as megaspore mother cell there. So it is megaspore mother cell. So even the megaspore mother cell, so the same nucellus cell is there. One cell of this nucellus tissue is working as megaspore mother cell. So then there is no much change is there. Only cell it will increase in size, and the nucleus will increase in size. Now it is the time of division. So it will undergo what? It will undergo meiotic division. So it will undergo meiosis, meiotic division. So then what happens? This megaspore mother cell, it will first form two cells, meiosis 1. It will first form two cells, meiosis 1. It will next form another meiosis. So it is in meiosis we have seen, in cell cycle and cell division we have seen there meiosis 1 and meiosis 2. So meiosis 2, it is similar to the mitosis there. The same we are going to discuss here. So it will form 4 megaspores. So it will form 4 megaspores here. So it is meiosis 1 and it is meiosis 2. So this is only one meiotic division, but I am writing twice because here meiosis 1 is taking place and here it is meiosis 2. So this whole is meiosis. So this whole condition is meiosis there. So when meiosis 1 takes place, two cells are formed. So these are dyad cells, megaspore dyad. So dyad will be in the form of what? What is the ploidy of these cells? So this is already here megaspore mother cell, it was 2n in condition. So when meiosis 1 takes place, it is a what? Equational division or reductional division? It is a reductional division where the chromosome number, it reduces from 2n to n here. The cells of dyad cells, it will be n in. The ploidy will be haploidy. The next meiosis 2 takes place. In meiosis 2, 4 megaspores are formed. So it is megaspore mother cell now resulting into tetrad or megaspore tetrad also we can call. This is megaspore tetrad. So when megaspore tetrad is formed, all the cells it will be in a haploid condition. All the cells of this it will be in a haploid condition. Haploid means a single set of chromosomes. So it is like a gamete formation. So in it is a megaspore mother cell which is having 2n. When meiosis will take place, these gametes are ready now. So it is like daughter cells, 4 daughter cells, it will be in a haploid content. This we call this as megaspore tetrad. Tetrad means 4, 4 tetra. So tetrad there. And each one it will be having n, n, n. So n, n, n. And do you know what happens in this case? In most of the angiosperms, angiosperms is nothing but flowering plants. So in most of the flowering plants, what happens is, so these three will get degenerate. Means these three will get degenerate, and after degeneration, so there will be only one cell left. So this only one cell left there. So this one we are calling this as megaspore, functional megaspore. This one is functional megaspore. So here 3 will degenerate. So 3 megaspores will degenerate. So 3 megaspore degenerate, only one will become functional, and this is, we call it as functional megaspore. And the ploidy of the functional megaspore, it is haploid only. There is no change in ploidy level. Only the change which is taking place here is only the 3 mega out of 4 megaspores, 3 are getting degenerate. Degenerate is nothing but it will dissolve the cell, it will destroy, and the molecules it will be dissolving that. That is 3 megaspore degenerate. And this one is a so this kind of development we call this as monosporic development. Monosporic development in the sense only one out of these 4 megaspore will become functional. So if only one megaspore is becoming functional means it is monosporic development. This we are calling it as monosporic development. Monosporic development in the sense out of 4 megaspores only one is becoming functional, that we are calling this as monosporic development. So you need to remember this point, it is very important. So this we are calling this as monosporic development. So in this slide, what we need to remember. So one is meiosis is taking place in one of the cell out of this nucellus cell. So one of the cell will takes place initiative and it will become. So it will come forward and it will tell. So I am going to become a class representative of whole class like that. So this one of the cell of this nucellus tissue, it will come forward and it will increase in this cell size and it increases in the nucleus and all. So it is going to function as megaspore mother cell. So this megaspore mother cell, when it undergo meiosis, so meiosis 1 will get 2 cells dyad and the meiosis 2 will get 4 cells out of this 2. So that 4 it forms, megaspore tetrad is formed. So when megaspore tetrad is formed, 3 is going to degenerate. Degenerate is nothing but, degeneration is nothing but there. It is going to dissolve. So the only one cell of this megaspore, it is going to become functional, that is functional megaspore. And this development is there, no, only one cell is going to become functional, that we are calling it as monosporic development. So this you need to remember, it is very important. So neat aspect, if you see, it is a monosporic development. In some cases, what happens, out of 4 megaspores, 2 will get degenerate and 2 will be functioning as functional megaspore. So in that case, we call them as bisporic development. Bi means 2, bisporic. 2 functional spore will be involving in formation of that, no? So that we call this as bisporic development. Whereas this one is monosporic development. So next we are going to see, now the megaspore is ready. Now the megaspore is ready, not a complete female gametophyte. So that, see, now where the megaspore is there, still in the within this one, means where it is still within the ovule there. Ovule, within the ovule, nucellus cell, one of the cell, it is turning into a megaspore here. So now we are going to see, this is about megasporogenesis. Now we are going to see development of a female gametophyte. So if you, we will be taking from the last slide only here. So continuation of this one, the continuation of this we will be seeing in this next slide. So the continuation of this functional megaspore, it will lead to formation of female gametophyte. I am writing here only, that is female gametophyte. So how the female gametophyte is formed, that the development of female gametophyte we will be seeing here. See, in the ovule we have seen there is a functional megaspore. So I am taking functional megaspore here. It is a functional megaspore. So this functional megaspore condition will be, the ploidy of functional megaspore will be haploid, because it has already passed that meiosis there. So it is haploid. So now what happens in case of this one? So I will write from the beginning so that you should not get confused here. So here nucellus cells are there. So here nucellus cells are there here. So one of the cell, it will be functioning from the micropylar end. That is very, very important. You need to remember that. So from micropylar end, one of the cell, it will be acting as megaspore mother cell. So this megaspore mother cell, see, initially there was what? There was nucellus cell. There was nucellus cell here. So which is diploid in condition. So one nucellus cell, it will become megaspore mother cell. So which is also a 2n in condition. After meiosis, 4 megaspores are formed. So out of that, 3 will get degenerate there. So out of 4, 3 will get degenerate and 1 will become functional megaspore. So this I am going to draw here now. That is functional megaspore. This one is functional megaspore here. So the functional megaspore is ready here. So this is functional megaspore which is haploid in condition. So in this functional megaspore what happens is continuous divisions are seen here. So this functional megaspore will undergo division. This one nucleus is there, no? This one nucleus, it will form two nucleus there. And the same one, continuation of this, another division will take place here. So these two nucleus, they will move towards the polar region. Polar means this is one polar end, this is another polar end. So they will be coming in here. And after coming here, they will be showing another mitotic division, another mitotic division. So there is another mitotic division has taken place. So in the next again, so two cells are there now. These all four cells, they will be showing another mitotic division. Here two, here four they form, and here four they form. So now four cells are there, four cells from there, that side. So this division is free nuclear division. You can see there is, so when the cells are, when the nucleus are undergoing division, I am not making any walls there. So here wall formation is not taking place, only nuclear division, free nuclear division, only karyokinesis is taking place. So here in functional megaspore, in the first case what happened, meiosis 1 has taken place here. So sorry, mitosis or mitotic division. So in first case one mitotic division is resulting into two nuclei there. In the second case another mitotic division resulting in four nucleus. So another case here another mitotic division. So totally how many mitotic division? 1, 2, 3. 3 mitotic division has taken place. Now 8 nuclei are there. So from this 8 nuclei, one will come from this end, this polar end at the center, and another one it will be coming from this polar end, from this polar end. So there out of 8 nuclei, so one will come from this end and another one will come from this end at the center. And all these divisions are there, no? So all these divisions are free nuclear division. So all these divisions from here to here, from here to here, all these divisions are free nuclear division. So it is free nuclear division. Without wall formation, free nuclear division has taken place, and this free nuclear division, 3 mitotic division resulted into 8 nuclei stage now, and there is still only one cell is there. So after 3 mitotic division what happens is, so after 8 mitotic division, so here you can see that is a free nuclear division has taken place. Now there is a transformation will be taking place. I am saying here 4 nuclei are there in this end and here 4 nuclei are there in this end. So in this end Micropylar end is there. This is micropylar end. This is callosal end here. So when micropylar end it is there, so three cells will gather together here. So there three cells are there, three nuclei are there here in this end. So there are three nuclei are there. In this end there are three nuclei. And I am saying from this polar end one nuclei is coming at the center, and from this polar end another nuclei is coming at the center. So it is forming two nuclei at this center now. So now the wall formation will take place. See, now understand this one. So now there is a wall formation is seen. So now there is wall formation is seen. So this is one cell, another cell, and this is another cell. Three cells, and this is another cell here. 1, 2, and 3. So three cells. So three cells at this end, three cells at that this end. And at the center, there are two nuclei are there. There will be no fusion of nuclei. There is no fusion of nuclei. They are separate. This is haploid, this is haploid. Because see here, functional megaspore, it was in haploid condition. So if haploid condition is undergoing mitotic division, this nuclei is also haploid, this is also haploid. Same, haploid, 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 haploid. So haploid, all these nuclei are in haploid condition. And here three cells are there, and here three cells are there. And at the center two nuclei are there. This one we call this as large central cell. This is large central cell. This is large central cell. So that is large central cell. Here what you are seeing, now it is time to label. So it is a complete female gametophyte is ready, and the process what you are seeing, the development of female gametophyte, or it is also called as embryo sac development also. This whole structure we are calling it as mature embryo sac, matured embryo sac now. So matured embryo sac has what? 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei. 8 nuclei are there. And how many cells? 1 cell, 2 cell, 3 cell, 4, 5, 6, and central one big large, that is 7 cell. So this condition, so 3 times they asked in PYQ that this is the embryo sac condition. 7 celled 8 nucleus. So there are 7 cells are organized and 8 nucleus, because at the center 2 nucleus are present in central cell. So that's why it is only one cell here. So this condition we are calling this as 8 nuclei and 7 cell stage. And this end is there, no? See, in the initial case we have discussed. So this in the ovule, micropylar end is there here. This is callosal end. Callosal end, and this is micropylar end. So this is micropylar end, and that is callosal end. See, initially there was free nuclear division where nucleus are undergoing free nucleus or dividing, just dividing. There is no wall formation. Only nuclear are undergoing the division there. There is no wall formation is not followed by it. So resulting until 3 mitotic division, there is no wall formation. After 3 mitotic division, wall formation has taken place here. So this is the development of a female gametophyte or development of embryo sac. So in the next slide, we will be discussing about the structure of embryo sac there. So here, structure of embryo sac. So this is embryo sac here, or the complete matured female gametophyte. So we know that it is 8 nucleate, 7 celled. So I am going to draw the same, but this time I am making chalazal end here. This is chalazal end, and this is micropylar end. This is micropylar end. So chalazal end has 3 cells. That 3 cells are this. So it has 3 cells there. So it is having 3 cells. That we call this as antipodals or antipodal cells. So 3 antipodals are there here. So these are antipodals. So at the chalazal end, antipodals is there. Remember always chalazal end, antipodals. So you can remember A and C. AC will be placing the upside, no? So that A and C will be together, antipodals at the chalazal end. And in the another end, you will be seeing synergids. So these are synergids here. So these are synergids. And one more is there, no? That is egg apparatus. So it is a main egg. This is egg. Egg and synergids together, we are calling this as egg apparatus. So that we call it as egg apparatus. And at the center, what you are seeing, the center, that we are calling this as central cell. At the center, you will be seeing that 2 nucleus are there here, that is central cell. So these are 3 antipodal cells, and the central cell, it is central cell, or which is having, it is central cell. And these two are there, no? These two, they are polar nuclei. So these two here, which are present, polar nuclei. So why we are calling this as polar nuclei? Because they came from 2 polar ends. So it came from this polar end, this polar end. So that's why it is polar nuclei. So it is polar nuclei which is present at the center, that is central cell. And the ploidy of this one, antipodal cells, all they will be having N, N, N. So all they will be having N. And the ploidy of central cell is N plus N it is. We cannot say it as a diploid, but it is haploid and haploid. 2 nuclei are not fused, but they will be together there. So fusion will take place later. Initially they will be, the 2 nuclei are just separated. That we call this as free nuclear condition, N plus N, that is central cell. And next is egg cell is there. Egg is also in N condition. Synergids, it is also in N condition. So all the functions of these cells are different. So that we are going to discuss here now. So antipodals are 3 are there. Antipodals helps in so these antipodals helps in one function is helping the nourishment to this one, nourishment to the developing embryo there. And one more thing is it is also helping them to guide the pollen tube there. So it also helps in guide the pollen tube. So one more structure I forgot here. So there is a finger like projections are seen here, that we are calling this as filiform apparatus. That we call this as filiform apparatus. This is, so upon the synergids only the filiform apparatus is seen there. So that next upon this filiform apparatus, so that it will be helping in micropylar end. No, micropyle is nothing but opening and opening pore. It will be how it will be. So if you see here the ovule, how it will be. So it is a micropylar end in the sense. So it is inner integument and it is micropyle, and this is outer integument. So when it is outer integument, same here in case of here it is also outer integument, and this end is micropylar end here. So this end is micropylar end. So through this micropylar end the pollen tube germination will take place and the male gamete will be reaching there. So this is egg is here, no, that's why it is will be micropylar end, egg will be present there. So there the pollen tube germination takes place and the fusion of gametes will take place there at that position. So here guide the pollen tube in a correct position there, and antipodals also helps in antipodal cells. They also helps in giving nourishment to the development there, giving nourishment to the developing embryo there. See, this is the embryo sac. Embryo sac means after the fusion of gametes, there the embryo is going to form. So that's why we are calling this as embryo sac. And female gametophyte is nothing but it is embryo sac here. So the condition of embryo sac is what? The condition of embryo sac is, that is 7-celled 8-nucleate cell. And the polar nuclei, it is in 2 n plus n condition is there, and this after fertilization. So this central cell after fertilization it is going to form primary endosperm nucleus, which is, it will be in a triploid condition. The ploidy of primary endosperm cell or primary endosperm PEC also you can call this one. So this is PEC, primary endosperm cell, which has triploid in condition. So endosperm it is going to form. So it is forming endosperm after fertilization. So after fertilization it is going to form endosperm cell. So the egg is there here. Egg is going to form zygote after fertilization. Zygote will form embryo there later. So embryo will form seed that later. Seed will form again a new plant. So this is egg. And synergids are there. Synergids also helps in guiding the pollen tube towards the egg there. But before that filiform apparatus are there. Filiform apparatus is also function is that to guide the pollen tube towards the synergids there. Guide the pollen tube towards synergids. So after delivering to the synergids, synergids will take care till it reaches to the egg there. So how it will be? So it will reach first, see the germination of pollen tube has taken place and the gametes are released here. So when the gametes are released, the filiform apparatus it will be guiding towards to move towards synergids. So after moving towards synergids, it will guide towards the egg cell there. So this is guiding or guiding the pollen tube, the male gametes towards to from here to here, filiform apparatus to synergids, synergids to egg cell. And one one gamete will fuse with this here to form 3 nucleus stage, that is PEC, primary endosperm cell, and another gamete will fuse with egg to form zygote and embryo is formed. So this is the complete embryo sac. So it is about the female gametophore. So we'll discuss some question answers related to this. The ovule of angiosperm is technically equivalent to, okay, you can answer here. Ovule of, it is a Neet 2016 question. So the ovule of angiosperm is technically equivalent to, what it is? Megaspore mother cell, not, because megaspore mother cell which is present within the ovule, megaspore is also same. Megasporangium, here it is the correct answer. Megasporangium is also equivalent, means it is also called as ovule there, megasporangium, not megasporophil. Megasporophil, where do you see? That in pteridophytes you will see, no? Megasporangium on the leaf, megasporophil. So it is option C will be correct answer. Choose the correct sequence. It is of 2017. They are screen which are there. So choose the correct sequence representing the ploidy of nucellus, megaspore, mother cell, and megaspore, egg cell, zygote, and apolar nucleus. They are asking about the ploidy. Ploidy means set of chromosomes in a cell. So nucellus is there here. Nucellus means what? Nucellus, the surrounding cells there. So that nucellus tissue, it will be in a diploid condition. So in this, three are there, diploid. The next, megaspore mother cell. Megaspore mother cell means, mother means it is in diploid condition there. Megaspore mother cell, diploid, it is not there, so we'll eliminate this now. Next is megaspore. Megaspore means, megaspore mother cell, after meiosis it forms megaspore. After meiosis, if it is forming megaspore, it will be in a haploid condition. So in both the cases it is haploid. Okay, done. Next, egg cell is there. Egg cell means after megaspore formation, there the embryo sac formation will be taking place, and the egg cell, it will be in a haploid condition, not a diploid. Then eliminate this one. The answer will be C here. 2n, n, we'll see further also. Zygote, it will be diploid condition, and a polar nucleus is n. I said no, polar nucleus will be in n condition, not a triploid. Don't go for this option here. It is n there. So triploid is not. So this polar nucleus, it will be in n condition. Next, polar nucleus of over. Secondary nucleus here. So that secondary nucleus is after fusion, that is 2n, and the primary endosperm nucleus, that is 3n. So the option C will be answered here. Next, a typical unfertilized ovary or unfertilized angiosperm embryo sac, at maturity it is having 8 nucleate and 7 cell cells. Okay, 8 nucleate and 7 cell cells, that is the correct statement. So the egg apparatus has 2 polar nuclei. So it is wrong, no? Egg apparatus will not be having 2 polar nuclei. So assertion is true and the reason is false here. So next, starting from the innermost part, correct sequence parts in ovule are. This is very somewhat confusing question. You need to analyze here. Starting from innermost part, the correct sequence of parts of ovule are. So innermost. So you need to come from, see, if it is ovule in the sense, it is ovule, you need to come from inner side in the sense from here to outer side like this. So which one will be the answer here? Egg is there. Egg, egg, egg. So they will be present inside of this one. So egg is there. So egg will be the first one, innermost. Next to that, what you are seeing, this is inner integument. Before inner integument, what you will be seeing, there is a layer of nucellus cells. No, there is a layer of nucellus cells. So egg, after that nucellus is there. Next to nucellus, what you are seeing, integument you will be seeing. Wait, embryo sac, integument and egg. Egg, integument and embryo sac and nucellus. So it is not because nucellus they are keeping the first last there. It's not. Egg, embryo sac, nucellus and integument. Okay. Here egg, embryo sac is there. So immediate to that, what you are seeing, this layer is embryo sac. Next to that, nucellus is there. So that is the second one will be the option. Egg, embryo sac is there. Next to that, nucellus is there. Next to that nucellus layer, you can see the integument. Second one will be the option. So next, what you are seeing, embryo sac, nucellus, integument, egg, they place it last. No. Egg, nucellus is given, embryo sac is given next. So it will be coming this way. This option B will be connected. Embryo sac is monosporic when it is developed from. So when we say that embryo sac is monosporic, so when we will be saying embryo sac is monosporic, when one will be the functional megaspore. So one of the four megaspore of a functional of a megaspore mother cell knot, one of the four megaspore develops from one of the four megaspores. So one of the four megaspores of megaspore mother cell. So we'll see the other statements also. Three megaspores of megaspore tetrad, no. Trisporic it will be. Two functional megaspores, bisporic it will be. The megaspore mother cell where meiosis has occurred but cytokinesis does not take place, it is not. So the option 1 will be the correct answer. So next, okay. So in the next session, we will be discussing about how the bringing together of both the gametes. Now the male gamete is ready, pollen grain is ready, and now the female gamete is also ready. So when both male gamete and female gamete, hero heroines are ready now. So we need to make them to meet. So that we will see in the next session. So until that, take care. Bye bye.